associated with molecular genetics or genetics. So the first terminology that we take is genetics itself. Genetics can be defined as a study of two distinct aspects. It is study of heredity and variations. The term heredity tells us the storage, expression and transmission of characters from one generation to another. So we have three events here: storage, expression and transmission of characters. From one generation to the another generation. So this is heredity. Whereas the variations are nothing but differences among the individuals of a species. So when you study heredity along with the variations, you have the genetic study of organisms. The variations are whatever the differences that we are seeing between two individuals, maybe one tall, another short, and so on, occurs either because of differences in genes. Then we call that as genotypic. You may also have a possibility the variations arise because the two individuals are grown in two different conditions. So environmental variations. Or there is a possibility, third possibility, we have both combined. There is a difference in the genotype and there is variation in the environment. So these variations in a long run can lead to speciation, and speciation can result in evolution. So that's the first term, genetics. It studies heredity and variation. Heredity is storage, expression and transmission of traits from one generation to another generation. The variations are the differences that we see among the individuals of a population or a species. We can have variations either because of genotype or because of environment, or you can have the two together forming the variation. These variations in the long run during the course of evolution generate new species, and ultimately the formation of new species result in evolution. The second term that we have in the genetics is the chromosome. So we have Sutton's theory. First proposed by Sutton and then modified by Boveri. They proposed chromosomal theory of inheritance. According to them, chromosomes are the carriers of hereditary factors. So the chromosomes contain hereditary factors, and these hereditary factors, which was a name that was given by Mendel, was later replaced with genes by Johansen. So chromosomes carries the hereditary factors. That's the first point of Sutton and Boveri theory. The second one, each hereditary factor or each gene is located at a specific position on the chromosome. And this specific position where we find the hereditary factor is called locus. So a chromosome is made up of several loci, each locus containing a specific character. So this was the chromosomal theory of inheritance that was proposed by the Sutton and Boveri, and for several years it remained without any challenge. But later on, when people identified transposable elements, we had exceptions for chromosomal theory of inheritance. The reason is transposable elements are relatively unstable molecules or unstable sequences of DNA that move from one position to another position. So they don't have a fixed place. But chromosomal theory says genes are fixed in position. If you define chromosomes, you can define them as self-duplicating structures. The self-duplication is what we call it as replication. Self-duplicating structures carrying genes. The chromosomes can be divided into autosomes and allosomes. These are the two broad categories of chromosomes. Autosomes are chromosomes that are directly not involved in sex determination, whereas allosomes are the chromosomes that are directly involved in sex determination. So autosomes, no direct role in primary sex determination, whereas allosome involved in primary sex determination. An organism contains several autosomes. For example, humans have 44 autosomes or 22 pairs of autosomes. When it comes to allosomes, usually you have a single pair. That single pair may be similar when you have cases like XX, ZZ, or you may have different if the two sex chromosomes are similar, we call such individual as homogametic. So human females, male birds, they are homogametic. If the two sex chromosomes are different, then we call individual as heterogametic. Hetero means different. So it's producing two different kinds of gametes. Half of the gametes here will contain X chromosome, half of the gametes here will contain Y chromosome. In this case, half of the gametes contain Z chromosome and the other half contains W. So here we have human males and female birds. So in birds it is reversal to that of humans. In humans, homogametic sex is female. In birds, it is male. In humans, heterogametic sex is male. In birds, it is females. So this is one classification of the chromosomes. The second classification of the chromosomes is based on their structure. We have homologous chromosomes and non-homologous chromosomes. When you have two chromosomes that are structurally similar and contain the same loci, then they are called homologous. So let us say this has a chromosome, this has a centromere, we have A, B, C, D. This is the second chromosome, has again the same genes. So these two will be homologous. Homologous chromosomes undergo meiotic pairing. And form bivalence, or because there are 4 chromatids, we call tetrads. So that's a feature which is important in terms of genetic recombination. So homologous chromosomes undergo meiotic pairing and then they form bivalents or tetrads, and these tetrads will get separated during the gametogenesis. The non-homologous chromosomes, they have no similarity and their loci are different. So let's say this is P, Q, R, S, T. Let's say this M, N, G, Y, X. So they don't have similarity in structure. One is acrocentric, another one is almost a submetacentric, and the loci, they are totally different. These chromosomes, non-homologous chromosomes, exhibit independent assortment. So we have crossing over taking place, recombination generated in both the cases. If you have homologous chromosomes, recombination occurs by crossing over. So that's what happens here. Whereas when you have non-homologous chromosomes, recombination occurs by independent assortment, where one gene independently assorts with respect to another gene. And these are the genetic recombination methods. So we have two different kinds of genetic recombination methods depending upon how the genes are localized. One method is crossing over method. This occurs when the genes are located on the same chromosome. The second one, independent assortment. This occurs when the genes are located on different chromosome. The homologous chromosomes undergo the crossing over. Non-homologous chromosomes undergo independent assortment. The next term in the terminology is the gene itself, a term that was proposed by Johansen. A simple definition for gene, which may not give anything, is it is a classical unit of inheritance. So even though you gave a definition, but really it will not tell anything except that it's a unit which is involved in the transmission of the traits from one generation to another generation. A better definition for gene is it is a contiguous, which means continuous, stretch of DNA that codes for an RNA. So that means this stretch of DNA undergoes transcription to give RNA. This RNA may be mRNA, then it can act as a template for protein synthesis. Or it can be ribosomal RNA, tRNA, which cannot be translated. Any gene which produces an mRNA so that the mRNA can be subjected to translation is called a structural gene. So a structural gene codes for proteins. If a gene is continuously expressed, then we call that gene as constitutively expressed gene. So there is no switching off, switching on for this. Once it is switched on, it just on expressing. So continuous expression. You have less regulation. You don't really need much of the regulation. If you have a gene or if you have a set of genes that are needed for the basic functioning of the cell, so that it has to be expressed in all the kinds of cells, then it is called housekeeping genes. So housekeeping genes are required for normal functioning of the cell. So in every cell we see the expression of housekeeping genes. Then we have a set of genes which are called smart genes. The smart genes are those that are expressed in either specific tissue so you can take the example of insulin gene, which is expressed only in the beta cells, or specific state of development. This we can take example of the beta globin gene of hemoglobin. So the expression of the gene takes place when there is a switching of hemoglobin from fetal hemoglobin to adult hemoglobin. Then Wenger gave the fine structure of gene. So until Wenger gave this concept, people thought you cannot further divide gene into discrete units. Wenger said a gene can be divided into 3 distinct regions. The first one is cistron. The cistron region is the largest region, or most of your gene is nothing but the cistron. And what the cistron does is, cistron codes for the protein. So cistron is the largest unit, cistron is the functional unit, it is the one which codes for protein. The second one is recon. The recon is unit of recombination. or you can say unit of crossing over. The minimum size of this is one base pair, that is two nucleotides. The third unit of the gene is muton, which is unit of mutation. So it indicates the smallest part of the gene that can be subjected to the mutation. The minimum size of the muton will be one nucleotide. So see the difference? This is one nucleotide, this is one base pair, that means two nucleotides. So depending upon whether a given gene contains only one cistron or multiple cistrons, we have two different kinds of genes. So the first kind of genes are monocistronic genes. So mono means single, cistron means only one functional unit. So genes that specify single RNA or single protein. So one gene, one protein or one RNA. It's common in eukaryotes. That's because eukaryotes have so much of genome that they can have considerable cistronic differentiation, and also they can have barriers to separate one gene from another gene. The second one is polycistronic genes. So poly means many. That means these genes specify more than one RNA or protein. This is something that is common in bacteria. In case of bacteria, the polycistronic genes are organized into functional units which are called operons. So when you take an operon, you have a regulatory gene here, that regulates the entire operon, a promoter region for structural genes, then you have an operator sequence, and then you have structural genes which code for proteins, and then termination. So when the operator is free, operator is binding site for repressor, promoter is binding site for RNA polymerase. So assuming that the operator is free from repressor, then RNA polymerase binds to the promoter and can transcribe this. So you get S1, S2, S3. So this will be the initiation codon for translation of S1. This is a termination codon for S1. This is the initiation codon for S2. This is the termination codon for S2. This is the initiation codon for S3. This is the termination codon for S3. So when the translation takes place, you get protein 1, protein 2, protein 3. That means the expression of a single gene produced 3 distinct proteins involved in the same biochemical pathway. So polycistronic genes specify proteins involved in a common biochemical pathway. So the advantage that you have in such cases is by switching on or switching off the promoter sequence here, you can either transcribe your structural genes and therefore you can get the proteins needed for a particular function, or you can stop the structural gene expression so that you don't generate the proteins which are needed for a particular pathway. So this is important in those cases where if the substrate is available, you can go for the synthesis. If the substrate is not available, you can stop the synthesis of the gene